Okay, um, everything that we know is historical. So something happened beforehand and we've learned from that, and we're in the present, but we have to somehow get the present into the future. And I think that one of the things that we're learning is that some of the concepts that Bill has highlighted and working in conjunction with Bill Weirder and other colleagues is where we are now from our understanding of the mechanisms of the disease, and now we have to bring some of these into the future. So about 15 years ago, I started to talk about the cure of CLL, and they said, there, I knew he was crazy, and he still is. But now we have the situation where we have a program which in patients that need treatment have a very high complete remission rate. That is that no matter how hard we look, we can't find 1 in 10,000 or 1 in 100,000 CLL cells. So that we call that PCR negative. It can't be found on polymerase chain reaction or a flow cytometry negative state. I was just checking the other day of the first group of patients that got started on the FCR program about 11 years ago. We now have 22 of these first group of patients that have been in continuous remission for 10 years. By the end of this year, there'll be another 9, so that we now get into the situation where we have a very large number of patients with very long remissions. We also now have different ways, including lenalidomide and a new antibody called Zera or ofatumumab that can potentially mop up these remaining leukemic cells without damaging the host. We talked a bit about the specific targets, and now that we know that there are specific targets, we may be able to identify very specific approaches so that we'll be able to treat the patients earlier because they'll tolerate the treatment better. They will have had less chance to evolve cells that are resistant, and as I've highlighted for you already, I think that the thing that's going to change our ability to cure the disease is to teach the immune system at the present time an allogeneic or someone else's immune system to kill off the cells that are there and keep on killing off subsequent generations. But Bill Weirder has already highlighted that we're in the process of educating our own cells to do that, so that we know that the CLL cell is a very good target, and we just have to make our own cells better at doing what someone else's cells do. So when I mentioned this PCR negativity, we now have the situation where a number of patients will still have a small amount of CLL left, and that's called minimal residual disease. And there are different ways that we may well be able to measure just in plasma, different adhesion molecules that get the cells stuck to these stromal cells of the microenvironment. These microRNAs, as the cells break down, they liberate them into the plasma. They're very stable. You can recognize them because they have a unique sequence. We can measure different parts of the cell that float free. So the treatments that have been given as alemtuzumab or Campath has been very good at getting rid of these cells, but it also gets rid of a lot of normal cells. So that creates some problems, and we'd rather get to a more specific approach, and the Zera or the ofatumumab and Revlimid are those. Bill Weirder also mentioned that lenalidomide is a way that we can build up the antibody production, the normal antibody production, and that's a good thing. We now have ways of incubating T cells together with little beads with stimulating molecules that can increase the number of T cells in a bag 100 to 1,000-fold. And they become activated, and you can transfuse them in, and they will stay active for a year or more. And these are the cells that we're eventually going to educate to attack the CLL cells. There are some more new targets. There's this protein from a gene called ROR1, and ROR1 is something that's present in most cells until the time of birth, and then it gets shut down. But it doesn't get shut down in the CLL cells, which makes it a CLL-specific target for treatment. We also have the B cell receptor, and this is the part of the B lymphocyte that recognizes an antigen or a part of, say, a virus or a bacteria. It lodges into this little B cell receptor and switches the cells on. In each CLL patient, every cell is an identical copy from the B cell receptor, and that there are about 15,000 families of these cells, but all of the CLL cells only have one B cell receptor. So again, this is a patient-unique target. So we have the B cell receptor now. We know that there are at least 4 important tyrosine kinases that activate downstream activity of the cell, and these are all in clinical trial at the present time. There's a fascinating drug called KEL-101 that inhibits P13I3 kinase delta, and we're now about to activate the first study of that combined with an antibody. And there was this guy, Bruton, years ago, who was a naval doctor, and there was a child from the naval base that kept on having repeated infections, and as time went by, they found that he couldn't make any antibodies and he couldn't make any B lymphocytes. And that means that this particular pathway is a crucial pathway for B cell development. This is already in the clinic. I saw 2 patients today. It actually melts away lymph glands very, very quickly. It pushes the cells out into the bloodstream, whereas Bill Plunkett mentioned they're very susceptible to either dying naturally or being killed by other molecules. Dr. Peng Wong, who's one of Bill Plunkett's students, has developed a method whereby we can make reactive oxygen species instead of protective to making them fatal molecules. And it's interesting, PEITC is the name of it, but there is also another derivative called selectosine, and both of these kill off aggressive CLL cells, including the 17p deleted, the p53 abnormal cells, better than anything we've ever done. And this is the molecule that I've been bothering you with in the Momentum magazine, from the watercress and the broccoli and cauliflower, etc., all those vegetables that our mothers told us were good for us, but we never ate anyway. But now we're getting to the point where vegetables or a chemical from there can actually kill cancer cells. So CLL is a very specific cell. It's a big cell. It's relevant. It can be attacked. The way that we've been attacking it in the past have been antibodies, and Campath, rituximab, Arzerra are approved. There's another one coming along called GA101, which works by a different mechanism. There are bispecific antibodies where one bit of it attaches to the CLL cell, the other to the T cell, and brings them in immediate proximity. And this is the same situation with CARs, that there are chimeric antigen receptors which can be put into the immune cells, and it brings them in contact with the leukemic cells and kills them, and it learns to keep on killing the cells. And we're very impressed with the multiple aspects of lenalidomide. So when we have these very specific molecules, many of them are now oral, they have a very favorable toxicity profile. Why shouldn't we treat patients earlier? Well, in the past, when we would treat patients, we would damage the immune system. So if we do the watch and wait, we're not doing anything to the genetically abnormal cells, so we have to balance out the risk and the benefit. So while we have taught from Osler that we should do no harm, but we should do good, and we haven't been doing good in this early incubation phase of the disease. So one of the things we're doing now is to identify the patients that are most likely to progress in the 1, 2, or 4 year time periods, so that we can actually give them the opportunity to benefit from these oral non-toxic preparations. Are we there yet? And the answer is, in some patients, I think we are. I think that some patients are presently being cured. I think that there are a number of problems. In CLL, the dysregulation of the immune system leads to antibodies which attack red cells, attack platelets, attack a variety of other cells. The large cell transformation, which I'm pretty convinced in many cases is driven by a number of viruses, where you have to repair the immune system. There is a tendency for CLL patients to develop second cancers, especially skin cancers, prostate cancer, and there's a general increase in other malignancies, so we have to be on the lookout for those, so that we should do all the mammograms and the pap smears and things like that, and the infection occurrence and prevention that Bill Weirder has already mentioned. But one of the things that's happened is that in that period of time, we have certainly doubled the survival of patients with, that need treatment for CLL. We have increased the number of 10-year survivors over that period by a factor of probably 10, from 5% to about 50%, so that there is no reason that we shouldn't be optimistic that this is going to continue.